Welcome everyone to our session today on doing good science when resources are limited. This is an event put on by the community for Rigor. We're happy to have with us as our group of panelists and experts, Dr. Stephanie Hicks, Dr. Michael Gianfrido, and Dr. Dan Kessler. I have a few, let's see. Oh, sorry, I should introduce myself. My name is Hao Yi. I am the curriculum lead here at the Community for Rigor. And let me introduce our speakers, and then we have a little bit of poll for you all before we get started. So let's go. Dr. Stephanie Hicks is an associate professor in the departments of biostatistics and biomedical engineering at Johns Hopkins, with affiliations with Data Science and AI Initiative, the Malone Center for Engineering and Healthcare, the Center for Computational Biology, and the Department of Genetic Medicine. She is an expert in developing scalable computational methods and open source software for biomedical data science, in particular single cell and spatial transcriptomics genomics data, leading to an improved understanding of human health and disease. Dr. Michael Gianfrido is a pharmacist and health services researcher with expertise in evidence synthesis, qualitative and mixed methods, and shared decision making. Dr. Gianfrido's research focuses on optimizing medication use and promoting patient-centered care through shared decision making and minimally disruptive medicine. And Dr. Daniel S. Kessler joined the faculty of the University of Pennsylvania School of Medicine in 1995 as a member of the Department of Cell and Developmental Biology. He is now Associate Professor of Cell and Developmental Biology, Associate Dean for Graduate Education, Director of Biomedical Graduate Studies, Chair of the Institutional Biosafety Committee, and Co-Director of Penn Prept. He served for 15 years as chair of the Cell and Molecular Biology Graduate Program. He has extensive experience as teacher and course director for graduate and medical students. Dr. Kessler is a recipient of the Jane Coffin Childs Memorial Fund Postdoctoral Fellowship and the Pew Scholar in the Biomedical Sciences Award for his teaching and mentoring efforts at Penn. He was recognized with the Dean's Award for Excellence in Graduate Student Teaching in 2009 and the Christian R. and Mary L.F. Lindback Foundation Award for Distinguished Teaching in 2019. He has served as a member of the board of directors of the Society for Developmental Biology. His research interests focus on fundamental mechanisms that establish the major cell lineages and organizing centers of the early vertebrate embryo. And we're going to launch our first poll for everyone, which is about your biggest rigor worries in science. So we're not gonna collect this data for any nefarious purposes. We just kind of want to kickstart the conversation here on how you do good science and what your concerns are. And as our speakers get ready to present their comments, I'm hopeful that this will give us some interesting perspectives to talk about. Okay, so I'm going to give you maybe 30 more seconds. Oh, you're right. There is no other option. I'm sorry. If you have an option that is not one of the options there, please go ahead and write it in the chat. Thank you for pointing that out. Okay, so we have ended our poll. I did see ethical use of AI has popped up as a concern, and institutions not really caring about rigor as a concern um, in addition to some of the other options. Actually, is everyone able to see the poll responses, or should I share that on my screen too?
I can see them how.
Okay,
Great. Thank you.
great. Any initial comments from our speakers here about these poll options?
I mean, I could certainly say that this covers a broad range of the frequently, encountered questions in project development and data analysis and so forth. I mean, for me, one of the recommendations that we make strongly is to, consult with experts at the beginning of a project to develop the statistical and power analyses that are required for a rigorous study. And so many of these classes of responses would certainly fall into that area of planning.
Yeah, how if I can comment here on some of these responses and some of the things I'm seeing in the chat. Um, there's a comment that institutions don't really care about rigor. Well, this initiative was funded by NINDS, so the parts of the NIH definitely care about rigor, and I believe new guidelines are out there from the NIH about making sure that you address rigor in grants. So I can't speak for other places, but we were funded by NINDS.
Yeah, great mention, Mike. Dr. Hicks, any initial thoughts?
Yeah, I mean, I think they generally also align with what I would expect. I like the distribution across all kind of like all of the comments, so that's interesting to see as well. So.
Yeah. I was a little worried when telling Kara to set up the option that people could select multiple options that we would just get a flat uniform distribution. So I'm glad that wasn't the case and got a little bit of concentration on what people were worried about the most. All right. So with that, I think we're ready to jump into the speakers to give their own perspectives. Mike, do you want me to share the slides that you sent along, or do you want to share yourself?
I'm gonna try to share myself because I made a few updates, but if not,
Okay.
there weren't that many updates. All right. Um, okay. Are you able to see the slides?
Do see your slides. Oh, yep, and now we see them full screen.
Okay, great. So I was already introduced, so I'm not going to bother with that. And I did see someone in the chat who does evidence synthesis as their work. So I'm glad I have at least one person in the audience I can speak to. But there are a lot of rigor trade-offs that happen in evidence synthesis, and so I want to speak briefly about that today. So for those of you who don't know, evidence synthesis is the systematic and transparent identification and synthesis of evidence, and evidence can be all types of things. It could be randomized trials, it could be observational studies, it could be preclinical studies, it could be qualitative studies, it could be a lot of different things. There's many different types of evidence synthesis, and there are over 1,000 different terms used for this thing. Um, and the evidence synthesis plays a big role in science, again, regardless of what your science is. It can serve to orient researchers to a field, summarize existing evidence, inform research practice and policy, hopefully prevent unnecessary replication and waste, and identify areas in need of further study, whether that's because there's a lack of studies out there. There's studies out there, but they're not directly applicable to your particular question. The classic example I tend to give is if we have studies in adults, but we're really interested in kids, right? And there's no studies in kids. There's some indirectness there because we have some studies in adults. Imprecision. So we might have an estimate of effect of something, but the confidence intervals, for example, are very wide. Um, Heterogeneity. We might have studies, but they're giving us different answers. And evidence synthesis can help us understand that. Similarly, evidence synthesis by pooling studies can help us uh, better get a more precise estimate of effect. Also, evidence synthesis can identify whether the existing evidence is biased or not. Um, However, just like any other design, evidence synthesis can have varying levels of rigor, and that lack of rigor can lead to inaccurate or misleading results. For example, retracted studies continue to be included in evidence synthesis, leading to bias estimates and have been used to inform clinical practice guidelines. And conflicts of interest are still quite common and can influence findings. I personally believe that every project should begin with an evidence synthesis. You really should understand, what's out there before you dive in. This is important, especially if you're not experienced in a field, to know well, what has been done, and you don't want to create redundant science because redundancy is common, not only in science but in evidence synthesis. So if you look and someone's done that already, you don't need to do another one. But you want to check, is that evidence synthesis well done? If not, maybe you need to redo it or it's out of date. There was a study several years ago that the median evidence synthesis goes out of date in about 5 to 6 years, but 25% may go out of date within 2 years. But this is very field and question specific. So I encourage you to do some type of some type of evidence synthesis. However, there are a lot of challenges. And the little asterisk there is that Gen AI tools may help, but development and assessment is ongoing. And as someone in the chat noted, there may be some ethical concerns as well. But challenges include it requires content and methodological expertise to just to understand what you're looking at. It's time consuming, resource intensive, and the evidence base may be large, diverse, and continuously evolving. So, if you do the work to gather the evidence, but by the time you finish gathering and analyzing it, there's more evidence, you're stuck in this cycle here. So that can be tricky. And there are a lot of trade-offs that happen throughout this process. A big trade-off is with scope. Sometimes you have to look at the question, right? Maybe you want to look at what is the impact of uh, beta blockers on heart attacks, and beta blockers are a type of drug. But there's lots of different types of beta blockers, and so looking at all of them, right, that might not be as feasible as something looking at a particular beta blocker, but that may be potentially less interesting. So there's a scope of your question that's broad versus narrow. Um, Also looking at the search. So when you're doing an evidence synthesis, you come up with a question and then you search the literature in a systematic um, way. That's what makes it science. And you can look at language. And so oftentimes, um, at least in the English speaking world, we tend to limit it to English. And the impact of that language bias is really question dependent. Um, but you might be missing a significant portion of the literature depending upon your question. And so the rigor of your answer may vary, right? So you have to be thoughtful about, do I include or exclude non-English studies, for example? And there are translation resources now that are widely available, whether they're Gen AI tools or more classic translation tools, and they may be sufficiently accurate, but again, they're not necessarily 100% accurate. Um, there's things like terms when you're searching the literature. You have to balance, do I want to try to get every single potential study? And I use very broad terms. And then you get a data set of 10,000 abstracts or 100,000 abstracts, but your hits are very few. Or do you want to get fewer hits, but they're more likely to be relevant, but you may miss other studies. And so there's a balance between, when you're creating your search, of having too much or too little responses and risking either it not being feasible or missing relevant studies. Lastly, on the slide, databases, right? You're trading off feasibility in terms of, well, I can just search PubMed, right? That's fine. Well, not everything is indexed in PubMed. And so maybe you would want to search multiple databases, but maybe you don't have access to those other databases. Or looking at the gray literature, things that are not routinely indexed in databases. Um, and so you might not have a comprehensive look in the literature, but your fees, what you are feasibly able to do, right? So there's a trade-off involved in that. There's also trade-offs involved regarding the methods. So ideally, you'd want to do duplicate reviewers and duplicate extractors as you're looking at the data, at the evidence. This helps mitigate mistakes and individual biases. I can tell you from doing many of these that, after looking at 1,000 or 2,000 or 10,000 articles, you might make a mistake. So it's helpful to have somebody check on that. Um, Also, but you have to have that other person, right? If you're working alone, you might not have that. And so, sometimes there's a trade-off. You might be willing to accept those few mistakes. Um, the other issue is author contact. And so, there's often times where um, they may say something in the methods that a piece of data they collected, or you might find a protocol and a piece of data they were supposed to collect, but it's not reported. And so you can contact the authors about that data, or if data is only presented in a graph or some other format, you can contact the author to get that data. Um, but that takes time, right? You have to email them, you have to wait for the response, you might have to follow up. That takes time. And not everybody does that step, but it can improve the rigor of your study. And a lot of these trade-offs, I had this note on the other slide as well. There's a subtype of review called rapid reviews, and they have made a lot of these uh, choices in rapid reviews. For example, having one reviewer or having one database, and there's a lot of evidence built around of rapid reviews and kind of those trade-offs. And so if you're more interested in these sorts of things, I have some references I can share. Um, but the whole point was of this sort of case study um, is that, I think rigor is key to good science. I think poor rigor can have real world impact. Um, as I mentioned in the beginning, understanding the need for a project. So in this case, should you do an evidence synthesis? If you're not doing an evidence synthesis, where does your individual project fit within that larger body of literature? Um, that can potentially help avoid the need to make those trade-offs. So understanding, in terms of evidence synthesis, do I need to do one? If you don't have to make those trade-offs for evidence synthesis. But in your projects, if stuff has already been done, maybe you don't have to do whatever your project is, and you can do something more focused or feasible, for example. But if you do have to do trade-offs, they should be made deliberately and thoughtfully, and being aware of what those trade-offs are and the limitations that are built in, so you can address those if possible. Um, and I'll just keep these up here for a second, so they're on the recording. But if people have questions, they can reach out to me. That's all I have for slides. And so I don't know if we're taking questions now or we wait till the end, Hal, but you let me know.
I think we're open for any quick questions or comments from the panelists. But if the audience, if the audience could hold their questions until all the speakers have had a chance to go, that would be great. I know, like, we want to make sure we have enough time for our speakers today. Maybe I will have a quick question for you, Mike, which is, I think when you are looking at studies to include in a systematic review, what are some, what's one thing you look at first, I guess, what's one sign that you're like, oh, this is a great thing to include, or this is a warning flag for possible exclusion.
Yeah, absolutely. Well, again, with the systematic, so with reviews, right? what makes a systematic review or an evidence synthesis different is there is a predefined protocol. And again, to do rigorous science, we need to make that protocol, but also follow that protocol. So part of that protocol is your inclusion exclusion criteria. So clearly defining what is in and what is out. And so, in terms of what gets in, it's very project specific. So I don't know that there's anything, unique, but I will say that certain databases and certain platforms now do flag if an article is retracted. So I think if you're looking at it in that window, paying attention to that and being mindful of, geez, if something's retracted, I don't know if I want that influencing the literature, unless of course you are interested in retracted studies. And there are several systematic reviews of retracted studies. So
Yeah. All right. Great.
Chris Olson has his hand up.
Hi, I just wanted to ask your thoughts about how do you think about this when you are writing a grant, where obviously this is a really important concept, but you have limited space on, and sometimes you have to address multiple pieces of subject matter. Everything from the rigor of the literature that's kind of aiming you towards your methods, and then also even the rigor of the actual methods you're using or some of the things you've already done in your own lab. But thank you for a great presentation too.
So I apologize, Chris, but I'm not sure I fully understand the question. But in a grant, I understand that there's space limitations. But maybe if you could clarify what your concern is.
Yeah, I guess how do you prioritize maybe the synthesis of information or even the citations you use to kind of, make the best use of that limited space?
Yeah, so I think that's where systematic reviews or evidence syntheses are helpful. Um, so instead of saying, this person said this, and this person said this, and this person said this, if you have a good evidence synthesis, you can say, overall, the evidence is mixed, or, it has these characteristics, and you have that citation. But by demonstrating that you have looked at the literature and have a good sense of what's in it. And again, it has to be a high quality review because, it's a like whole classic garbage in, garbage out, issue. Um, so making sure it's a high quality synthesis and showing that, no, we looked at the literature, here's the gaps based on a high quality evidence synthesis of the literature, and this is how we are going to address those gaps. And I don't know the most recent numbers, but historically, very few projects justified themselves based on a systematic review of the evidence. But thank you for your question and happy to follow up later.
All right. Thanks. We'll continue going on with Dr. Hicks. I don't know if you have slides you want to present or you are just going to speak out in the open.
Yeah, if that's okay. I don't have slides. I thought it would just be interesting to share a little bit about my experience as what's called an associate editor for reproducibility for a journal at the Journal of American Statistical Association. So I put a link in the chat now, which is kind of a summary of what I'm about to say. And I will also add just at the end a little bit about my own personal experience as an associate editor of reproducibility, but I'm also happy to take questions beyond the short few minute presentation, just kind of sharing my thoughts. So for those that are not reading along, a little bit about the background itself. So this was a pretty cool initiative that was started in 2016 by the Journal of American Statistical Association. I'm just going to call it JASA for short. And its aim was to address what we all call the reproducibility crisis, and which has been around since 2015. It has a lot of terms that are used interchangeably, such as replicability, reliability, robustness, many different things. And, why would the journal be interested in caring about reproducibility? I think there were three main reasons. one, reproducibility research really fosters a culture of transparency and accountability. It also enables trustworthy knowledge to be translated, where researchers can really build upon the backs of previous work, feeling confident about that. And then also, from a journal's perspective, a journal can really gain credibility when you hold researchers accountable for producing reproducible work. So I think the journal, at the time in 2016 recognized one, the reproducibility crisis, two, all of these reasons why it would be valuable to participate and to be able to make a stance more formally. So what happened was the initiative led to the formation of what's called the Associate Editors of Reproducibility. And that has been very fun to participate in. So I've been participating in it since, 2019, I think, or 2020. It was right around the pandemic. And in fact, we've done a lot of things, and I'll tell you about the initiative in just a second. But one of the things that I'm most proud of is in 2023, we actually formed a new award called the Reproducibility Award for JASA. And here, the goal was to try to motivate the people who are publishing papers, in particular students and postdocs in this journal, with an actual cash prize. So in addition to not only reproducibility being a main thing that's being evaluated at the time of publication, we wanted students and postdocs to really be motivated to be able to have this as a line item on their CV with a $500 cash prize. So lots of things have happened over the last few years. The next really important thing I want to mention is this idea of being a partner of, as opposed to an officer of, reproducibility. So all of the reproducibility editors view themselves really as a partner of a partner with the author who is publishing their work. We really want to make sure that and help ensure that the work that's being published in the journal is being able to be reproduced by others for all the reasons that I described above. We're not interested in policing or really trying to make sure that everything is reproducible. We want to try to guide the authors and provide resources to really enable reproducibility as much as possible. Reproducibility is very much a gray scale. There's no right and wrong, or that's our view at least. So it's really important that we come at this trying to be partners with the authors. I think that's a really important thing that I wanted to mention. From a practical perspective, the way that the reproducibility review works is that we've established and created a document called the Author Contributions Checklist, the ACC form, as it's colloquially known to me. And the form is really to document all artifacts about the data, the methods, the code, anything that the author is putting forth as, the novel contribution, the code to be able to reproduce the data analyses and all of that. So it's a document that the author themselves fills out, though the target audience for the document is really a future reader of the document who's interested in trying to get to know what was done by the authors, the original authors of the manuscript. So in fact, it's actually intended to be phrased in the present tense. So really trying to think about who's going to be reading this document in the future. In terms of a typical workflow for how this works, the workflow would be the associate editor reproducibility starts with the ACC form and checks for completeness and clarity, making sure that it's filled out. Usually the author has provided some kind of README in a markdown language or a text document, helping to kind of try to walk the reproducibility editor through what they have done. If not, that's often a first comment that an associate editor of reproducibility gives feedback on of, can you please provide a README to your reproducibility materials to help guide us through it? And then I would say the next thing is that the associated reproducibility just kind of looks through the directory structure, will look at the code itself, check for things such as, are you using absolute paths versus relative paths, or like have you hard coded some path to someone's computer, or thinking about documenting code. So sometimes researchers haven't documented any portion of their code, and so an editor would say, can you please provide some documentation kind of explaining what's going on in this function here? I can't say that I read every line of code. It's more of I just kind of point and click and look through, and kind of ensure that it looks like there's a sufficient amount of information that's there, again, with the intent of a future reader, hopefully wanting to make their lives a little bit easier. Sometimes the editors will actually run example code. There are various reasons why it's not possible to run every line of code. One reason would be installing all of the packages and the time it takes to actually run algorithms. Sometimes they can take a long time. That is not our goal. And so it's kind of an important point of discussion that I'm happy to have after the presentation. So we do not run every line of code. It's more of just kind of spot checking and looking around and thinking about what would make a future target user of this document and this code easier to enable more reproducibility in the future. And then the AER, the associated reproducibility, will just kind of summarize their thoughts and write it up similar to the way any a reviewer would of a manuscript. Again, really focusing on not the scientific content or the evaluation of the methods, but just about the reproducibility aspects of their work. In the paper that I linked, there's a bunch of do's and don'ts. There's an FAQ of the kinds of top questions that we get as associate editors for reproducibility. I will probably end my little discussion here, but if anybody wants to chat more about it, very happy to. So thanks.
Thanks. That sounds like a great initiative. Do we have any questions from the other speakers? I'll throw one out for myself. I know that different fields kind of have different approaches to reproducibility. I'm wondering, like, to what extent have the initiatives you all have created at JASA drawn inspiration from or looked at kind of like standardization with things like reproducibility packages that are required at some journals in like economics or political science?
Yeah, that's a great question. There is a lot of active area research, as you're saying. And I would say even just getting to this point has been kind of a real win for the statistics community. So, for example, one of the main motivating reasons that the initiative was started was that in 2015, they took a hard look at the amount of code or anything, any kind of reproducibility materials that were provided. And it was like less than 20% of manuscripts were submitting anything. And I think that tied again with the reproducibility crisis that came out around 2015, or that was the term introduced was in 2015. I think that was a real like pause point and motivated the journal to say, no, we want to do better. And so getting to this point where we have identified people to volunteer their time to participate as a reproducibility editor, and also building the infrastructure for how this all works with the journal integrated with the scientific review process. Again, not trying to hold up the scientific review process, but just working with the partners to say, okay, your paper's going forward. Let's just make sure your materials that you're submitting are as good as they can to really help ensure more reproducible work for the future users of your work. We do not require any kind of benchmark or workflow or package to be used, like the targets package or anything like that. at minimum, there needs to be like a README and things like that. And I'm happy to talk more, but I think it's something to think about for the future. But even getting to this point and having all the authors of the field of statistics have this as a norm now was like a big shift in the field over the last 10 years. So it's been a nice change, but there's still a lot more to go.
So Stephanie, can I ask, how has this been received by the community? Are people embracing this as a way of strengthening their own work and the work of the community at large? Have you found points of resistance?
Yes. So, yes, and yes. I would say that overall, it's been a huge win. But of course, there are people who wish to not have to submit anything because they feel like it slows them down almost. But for all the reasons that I described before, I think the 90% of the field feels this is like a positive win. And then students and postdoc that I've talked to over the years, there's always like a point of pride about thinking about, oh, I want to use, I want my reproducibility materials to hopefully be up for some kind of award. And so that was kind of our one carrot that we wanted to add to help motivate students and postdocs as they're submitting their papers. I Sometimes, we get feedback that this is slowing down the process. We really do not try to require like any minimum kind of thing outside of like a README and thinking about enabling more commenting of your code. There's variation among associate editors of reproducibility for sure, and that's something that we're working on standardizing. But overall, I would say it's a really big positive win.
All right, thanks. And then Dan, I think we're ready for your remarks.
Okay, great. Thanks, Hao, and thank you all for engaging in this discussion. So I'm going to take a slightly different lens to this topic, which is kind of practically how do we manage our goals around rigor and project design and the interactions with collaborators and mentors and so forth. I'm coming at this from the perspective of running educational programs in PhD education, and just decades of talking with trainees about some of the challenges that they faced that relate to the rigor of the work and the projects that they're doing. And so I'll just sort of organize this kind of in three parts. And the first part is really focused on how do you prepare to do rigorous work. So for the trainee population in particular, where there's a power dynamic and with faculty mentors, and also for many who are new to a field and new to the model, there's frequently a tendency to just rely on more senior members of your team in defining what constitutes rigor within a particular field. And I have seen many circumstances where that will eventually lead to difficulty in carrying the project forward and to completion because the trainee in those cases is not in a position to really assess the rigor of the work at the early stages. If they are relying on the perspective of others. And so, one kind of approach to this in preparing for rigor is if you're new to a field or you're new to a methodology or a model, is to intentionally put some time into, learning what are the standards for rigor in the field. How are critical reagents validated in your systems? If you're receiving critical reagents from others, are you in a position to actually do your own validation? And if so, to kind of learn what's needed to do that. If you're not in a position to do your own validation of reagents, cell lines, animal models, really seeking out those who have provided those reagents and asking them to give you sort of a record and history of the validation that they've done, so you at least have a sense of the depth of those efforts as well. when it comes to cell lines in animals, doing some preparatory work around are there known issues with propagation or matings and so forth? Are there potential known issues around stability of phenotypes and characteristics, right? Frequently, what we see happening is somebody will get an animal model from a collaborator, and over the course of months, we'll rediscover what some of the limitations of that model are, and which greatly impacts the rigor and the ultimate success of that. And so front loading a lot of thinking around that is critical, not only for the success of the project, but also for your advancement towards independence within your project and within your field and as a scientist. And so for me, I will tell you, I was a graduate student in the late 80s, right? We had DNA back then. We didn't have much more than that. And I will tell you that rigor was not a concept that was talked about or thought about at that point. And it was all sort of received through lineage. Whatever the standards of the lab were, those were the standards that you live by. And there was not sort of a more broadly defined set of community standards around that. And so, if you're a first-year graduate student or you've just joined a thesis lab, so make it part of your training experience to really dig into what rigor means within the field. And I see a comment that it's still like that, and that is absolutely true. But we can all make a cultural difference around these things if we engage in thinking about rigor ahead of time, and if we hold each other up to these standards. An additional part of this, which I mentioned earlier, is consulting with experts around statistical approaches and power needed for analyses, and really bringing in outside people with the relevant expertise, so that at the project development stage, you've done your very best to sort of build in rigorous approaches and so forth. Okay. So let's transition to the doing of the project, right? And so we like to think, let's just take publications as sort of the primary deliverable at various stages of career around the work that we do. And there's the paper we hope for, and then there's the paper we can actually have, right? And those two things are almost always different, right? And so as we enter into the project with great ambition and using cutting edge technology, and with well-defined hypotheses of a variety of them, almost as soon as the first experiment is done, we need to engage in a process of reassessment and revision and changing of the specific elements of the project and the approach and the methodology and so forth. One of the things that I see both from trainees and from faculty is that they love the initial concept, right? They're very faithful to those initiating ideas, and it can result in a certain inflexibility. It can be collective, right? It can be the entire group of collaborating people are just, they love the idea, they love the hypothesis, and they're just going to keep banging away at it, even when evidence starts to suggest that maybe changes need to be made. A second scenario, which is quite common, is that members of the team start to diverge on the strength of the original concept and maybe the rigor of some individual approaches and so forth, right? And what I often see is that while those concerns can percolate up early, the conversations often are delayed, pushed back, and it's not till rather late in the project in some cases, especially with junior members of the team who have concerns, that there's a delay in raising these concerns. And one of the things that's absolutely true, and this session is built around limited resources, which means limited funding resources and limited time, right? And often time is a very critical factor here, is that if you delay addressing concerns about rigor until later in the project, you're just giving yourself less flexibility and less room to move in making those adjustments. And so what I suggest then is that as part of your regular check-ins on the project, your one-on-one with mentors, your lab meetings, and so forth, that you make an intentional assessment of the rigor of the different elements of the project, right? Is this animal model, you know, giving consistent reproducible results? Is the phenotype sort of variable in a way that was not anticipated, right? So addressing those things up front and kind of flagging things that could perhaps undermine the rigor of the study and making that part of the conversation early on, I think is essential because you're setting the foundation for making beneficial changes, perhaps a little bit further into the project, as opposed to, let's say, going for a year with a model that's giving you variable results and not really raising those concerns. In some cases, the solution to variability is just do it again, do it again, do it again, and people can invest a huge amount of resources and time, going well beyond the point where they can certainly point at problems that may require an adjustment in the design as well. And then the third piece here that I want to comment on is when there's a divergence of views on rigor and the significance of the data as it relates to the rigor of the study. How do trainees go about navigating that, right? So I certainly have seen plenty of cases where a trainee will be banging away in an experiment and not getting sort of the clarity and the rigor of the outcomes, and will go to their mentor and raising concerns about the model or the approach or so forth, and the mentor will, faithful to his or her original ideas, will say, no, we're good. Just do it again, do it again. And then the trainee gets caught up in this kind of repetitious, effort, perhaps using reagents or a model that are just not capable of giving the needed outcome. And so what might one do under those circumstances? And this applies to trainees. It applies to collaborators. There's always a power dynamic there. There's always somebody who's going to take who may take a position of seniority in a way that could perhaps discount the concerns of more junior members of the team. And so, for example, for PhD students, one of your key resources will be your thesis committee, right? Right. This is a group of faculty who are specifically tasked in assessing the project design and the results as they go along. And often students feel like, well, there's sort of this high stakes meeting once a year, and they have to clear the bar. And if they clear the bar in that meeting, then they're good for another year. And they keep their distance from the committee because it's viewed as purely an assessment, sort of a gatekeeping function. But when students are using their thesis committees well, there's a more organic, ongoing discussion, seeking out the expertise of individual members of the committee, going to them with concerns about a method or approach. And here, I'm keeping my comments within the realm of good faith differences in opinion, not research integrity. Certainly we could hold sessions on those as well, but even with good faith differences in opinion, it's critical to take your concerns to trusted colleagues, trusted mentors, and just to get additional perspective on, how you're assessing the situation. And is my mentor's point of view, is it reasonable under these circumstances? Is there a middle ground here? Or do I need to really advocate for a change of approach or a change of model based on sort of systematic factors which would undermine the rigor of the study? And so there's an important aspect of continually aligning and realigning expectations around the project design, ongoing discussions about the rigor of the study as it develops. And also, maintaining good and productive communication around these issues as well. And then, the final thing I'll say is that each of us, in pursuing science of integrity, rigor, transparency, reproducibility, we have to be willing to give up on our initial idea of what the paper and what the project is, right? We need to be able to make adjustments. We may need to let certain goals of the project go if we find that they cannot be done in a rigorous manner. We may need to adjust approach, adjust model. We may need to limit the scope of our conclusions and qualify them, and maybe talk about things that we believe could be true but are not fully supported by the data, and being very honest and transparent about that. Different standards going into a peer-reviewed article as opposed to your thesis, right? The thesis gives you more room for speculative discussion of results and so forth. And so just creating as part of your training a personal focus on developing your sense of rigor and what the appropriate approaches are and so forth, and then continually reassessing and communicating with partners in the research endeavor to make sure that the plan is continually adjusted to maintain high-level rigorous work is an important approach to take. That's all I got.
Thank you, Dan. Any questions or comments from the other speakers?
I'll just say, this was fantastic advice, Dan, and I think all of the listeners and grad students should really take what you said to heart, because I think it was all really great advice.
Thank you.
Yeah, thanks. I especially liked what you said, Dan, about being intentional about reassessing and reevaluating after some of the initial data comes in. I do think that reflective practice is, and that kind of like iteration is a willingness to acknowledge that we won't know everything ahead of time and can't plan for everything ahead of time and may need to adjust. And I'm kind of wondering if, if you came to that through any formal training, or if you have ideas on like how students or trainees could develop that kind of practice.
I mean, for me, I've learned most of this through the experience of trainees, right? If I look back on my own PhD work and my postdoctoral work, you know, we were sort of doing things at the standard of the field at the time. If I look back on it now, there were strengths and weaknesses to the rigor of it. In my role as a graduate group chair and now the associate dean role, I have a large data set. Hundreds of students, studying for the PhD, and frequently coming to me to discuss, challenges on the project around assessing the rigor of the approach, interactions with mentors that at times raise concerns. And so I've been able to see it in all of its kind of facets and the myriad variety of it. And in coaching students through those situations and helping them develop ways to advocate for their own views of what the best science can be, what you heard from me really comes from hundreds of those interactions over the year, and sort of continually being able to assess the relative success of the advice that I'm giving the students, right? And kind of refining it over the years, right? Right. The goal here with trainees is to empower them to take a position, a strong sort of intellectual position around rigor, and to tell them that they're allowed to advocate for better science at all times, and that and then when those interactions get difficult, just to help them understand how they can recruit allies and how they can use effective communication in navigating those situations.
Great, thanks. It's definitely a privilege to receive, I think, your wisdom that has built up over all the experiences you have had and your own reflections on it to think about what processes could be improved or instantiated to improve science. We do have time for a few audience questions. If any audience members have a question, if you are willing to use the Zoom function to raise a hand or type it in the chat. I think, depending on, how many people we have, you could even try unmuting yourself. I don't know how many we have that will try to mute themselves at the same time, but I'm willing to give it a try and then reassess after collecting the initial data.
So
Looks like we
how
have.
yeah, I see a question from Emma in the chat. I'll read it for the record. When doing systematic reviews in an area that you have been studying for a while, do you explain, and if so, how the fact that you look at the data already before creating a protocol? In most contexts, it is better to not have seen the data and to not know the outcome. But if you regularly read the literature, you might have a general idea what's out there already and could accidentally bias the protocol. Similar question for how to explain when working with public databases that many people have published work on, so you have some sense of the contours of the data when designing your protocol. Um, so um, I definitely think, um, you know, again, I believe in doing a systematic review before doing a project, again, so you're not doing wasteful or redundant, um, work. Um, and so I think, figuring out how your piece of science or question fits in the literature, is important. But I do understand this idea of not wanting to potentially, bias the protocol. And so, sometimes that, you sort of have to, I think in qualitative research, we call it like bracket out or say, okay, well, what has been out there and kind of try to put that aside, um, and think about, well, how do I think about this potentially differently than what's out there? Or how does my idea sort of fit within that? Um, I'm not sure I'm asking or answering the question appropriately, but I don't know if Dan or Stephanie have another thought. Or how to, I guess you're part of this.
Yeah, I mean, I interpret an example of the question as thinking about like setting inclusion exclusion criteria about certain kinds of methodological practices of the papers and the that you would be including in your evidence synthesis, and then of course that being informed by past experience or domain expertise, and then being concerned that well that is a room that is a decision where there is the potential for bias to be introduced.
And I'm thinking about this more, and there's I forget what the term for it is, but oftentimes fields will go in certain directions, and then all of a sudden it'll shift to a different direction, and so oh, everybody's been doing this. This must be the way to do it. Let's do it that way. But oftentimes some of those seismic shifts happen because someone has taken a different direction. Um, so I think in terms of being biased by the literature, don't be afraid to think about things creatively. Um, and identify potentially, that as a gap.
Right. Well, we are just about a minute and a half from the hour, so I want to make sure, we don't keep our speakers past their dedicated time. So I do want to thank everyone for attending. Thank you to our speakers. I do want to highlight that C4R is going to send out another call for public beta testers for our unit on confirmation bias. So I think if you're here, you are probably on our email list and you can expect to see that email shortly. And then I think it looks like Dan is unmuted and maybe also has some final words to contribute.
No, I just wanted to, thank everyone for engaging in this discussion. I want to thank Mike and Stephanie. It was really interesting to hear their perspectives on this.
Thank you for the invitation. It was fun to be here. Take care, everyone.
Yeah, same. Thanks, Dan. Stephanie.
And thank you, Hal and Caro, for leading this effort. Super valuable.
Yeah.
Thanks. Appreciate it. You're welcome. And then Cara and I will make sure to get that recording link also sent out to everyone because we know some of you might be interested in some of the references and papers that were shared, including the comment on reproducibility at the Journal of the American Statistical Association. We'll make sure to add that link as well. So thanks everyone. Hope you enjoy the rest of your day.
Thank you all.